Plasmolysis. The behavior of the plant cells or tissues with regard to water movement depends on the surrounding solution. If the external solution balances the osmotic pressure of the cytoplasm, it is said to be isotonic. If the external solution is more dilute than the cytoplasm, it is hypotonic. And if the external solution is more concentrated, it is hypertonic. Cells swell in hypotonic solution and shrink in hypertonic ones. Plasmolysis occurs when water moves out of the cell and the cell membrane of a plant cell shrinks away from its cell wall. This occurs when the cell or tissue is placed in a solution that is hypertonic to the protoplasm. Water moves out. It is first lost from the cytoplasm and then from the vacuole. The water, when drawn out of the cell through diffusion into the extracellular outside cell fluid, causes the protoplast to shrink away from the walls. The cell is said to be plasmolysed. The movement of water occurred across the membrane, moving from an area of high water potential to an area of low water potential outside the cell. When the cell or tissue is placed in an isotonic solution, there is no net flow of water towards the inside or outside. If the external solution balances the osmotic pressure of the cytoplasm, it is said to be isotonic. When water flows into the cell and out of the cell and are in equilibrium, the cells are said to be flaccid. The process of plasmolysis is usually reversible. When the cells are placed in an hypotonic solution, which means higher water potential or dilute solution as compared to the cytoplasm, water diffuses into the cell, causing the cytoplasm to build up a pressure against the wall that is called turgor pressure. The pressure exerted by the protoplasts due to entry of water against the rigid wall is called pressure potential. Because of the rigidity of the cell wall, the cell does not rupture. This turgor pressure is ultimately responsible for enlargement and extension growth of cells.